dear learners, myself Bhavani Pentela, working as an assistant professor in Noida Institute of Engineering and Technology, Greater Noida. Today I am here to discuss the subject Pharmacology-I with the subject code BP404T, second unit with the lecture number 20. So, in this lecture we are going to discuss about the drug discovery and development and with the preclinical trials. Without any further delay, we will move into the class. Okay, so before discussing about the drug discovery, let me see what we had discussed in my previous lectures. So, in previous lecture we had discussed about the pharmacodynamics, which includes the receptors. We had discussed about the receptors theories, classification of receptors, and we had discussed about the most 4 important types of receptors like Z protein coupled receptor, ion receptor, enzyme linked receptor, and finally nuclear receptor. After of that we have a small look on dose response curve, therapeutic index, drugs modifying the action, what are the things that modify the drug action, factors that modifying the drug action. After of that we had discussed about the drug interactions, which include both pharmacokinetic interactions and pharmacodynamic interaction. After of that lecture we had discussed about the adverse drug reactions, we had discussed various types of adverse drug reactions, and in this lecture we are going to cover about the drug discovery and development. So, what is any drug discovery? So, drug discovery, it is a process by where you can invent or by where you can discover the new drug into the market. So, what it means? So, what is the main aim of this drug discovery? The drug discovery, the main aim was to identify the new compound, to identify the lead compound, which you can call as a drug discovery. So, in drug discovery process, it involves the identification of lead, it involves the synthesis, screening, and finally pharmacological efficacy by checking the with various assays. So, once the compound has been identified by the drug discovery process, then drug development will be started. So, what it means? First the process was drug discovery and later on it will move into the drug development. The drug discovery process, we will invent the new drug, we will discover the new drug, the new compound, we will identify the new compound which can be used therapeutically. And to identify this compound we can use lot of assays, or for example you can use the bioassays by where you can determine the pharmacological actions of any drug, you can determine the efficacy of any drug. So, we will check, first you will identify the new compound and after of that you will move into the drug development process. So, in drug discovery we will discover, so if you are talking about the last 50 years or last 100 years, very few drugs has been identified and came into the market and lot of drugs has been withdrawn from the market. So, the reason behind is because of its therapeutic index was efficacy was low or because of high adverse drug reaction of any particular drug which can withdrawn from the market. So, to discover a drug we having mainly 2 sources. We can discover the drug by the natural source or we can discover the drug by the synthetic source. The source for the drug discovery by the 2 process, one is natural source and second one is the synthetic source. So, what is the main aim of drug discovery? So, the main aim of drug discovery here to develop the clinical efficacy of any drug. So, what is the main aim here? So, what is the aim, what we are doing, why we are discovering the new drug, why we are discovering the new compound into the market? Because the main aim was to increase the clinical efficacy of any drug and we have to develop the safety of any drug. So, you had invented the drug, you had done all the parameters, and if the drug is not safety, then no one is going to use the drug. So, the second most aim of the drug discovery was we have to check the safety of any drug, and economical viable is also very much important in the drug discovery process. And finally, the drug whatever you are going to invent, it should be a new class of any drug. So, these are the most important aims to discover the new drug. And next, what are the challenges for the drug discovery process? Because yes, exactly you can say that this is a very big challenge to discover a new compound into the market, because it is not at all an easy process. It is very time taking process and it is very much economical in sense which can you have to invest a lot of money. It is most costly to invent a new drug into the market. So, limited success rate is also one of the challenge in this drug discovery process, and we need the skilled person to perform the research R&D trials for the coming process. And the most important thing was ethical challenges. So, here you have to take the animals, you have to take the clinical trials. So, you have to go with the human beings, you have to take the humans. So, this is one of the most important challenge to discover the new drug. And last most one is regulatory approval. So, you need the approval. So, you had discovered the drug, you had taken the ethical committee, you had having the skilled person, you having the money to invent the new drug, and finally, you need the approval from the regulatory authority. So, these are the main challenges which comes under drug discovery process. Okay, next, drug development. So, we had discussed in the previous slide what is any drug discovery. So, from this slide we will discuss about the drug development. So, what is any drug development? Once you had invent the drug, once you had discovered the new compound, and after of that the drug development process will be get started. First you have to invent the new drug, you have to discover the new drug, or you have to discover the new compound, and then the drug development process will be start here. And it is the process beginning of any drug which will introduce into the pharmaceutical market. So, it is in a process bringing a new compound into the market. So, drug development, it is a process where you can bring the new compound or new pharmaceutical drug into the market. Once the lead compound has been identified by the drug discovery method. So, in drug discovery method you had identified the lead molecule, and when this process has been completed, then the drug development phase will get started. And this drug discovery and development is not at all an easy step or easy process, because it is very expensive to develop a drug or to discover a drug because of high cost of R&D, because the high cost of research and development, and because of the clinical trials we are going to check the drug to the human beings. So, these are all very costly procedures or very high in cost. So, this is the reason the drug discovery and development is not at all an easy task to market the drug in, to discover or to develop the drug into the market. So, it will take most of the cost and most of the time to invent the drug. And next, if you are talking about here every pharmaceutical, so we have, if I am talking about the drug discovery process is not in a simple way. So, if you have to invent the drug, it taking a lot of years, almost approximately more than 15 years, or you have to spend lot of time, or you have to spend lot of money, or you need the very skilled person there. But we having a lot of industries, we having the research centers, we having the institutes, we having the universities, but all the institute, universities or pharmaceutical companies will not bear to do or to discover the drug, because it is a challenge. So, every university or every college or every research center or every pharmaceutical industry cannot develop the drug discovery program. So, develop the drug discovery program, it will take approximately 10 to 15 years or more than 15 years depending upon the drug which you are going to invent, or which the disease you are choosing for the particular drug. And the drug development process is typically, so here we am talking about the drug development process. In this drug development process, once the drug discovery has been completed and you had identified the new drug, and now the next step was drug development. So, in drug development, and compared to the drug discovery, the drug development phase was somewhat very lag. It means it will take a lot of time to develop the drug and to check the parameters whatever necessary to the particular drug to go with the clinical trials. So, in this drug development phase, it has been classified into mainly two major steps. One is preclinical development and second one is clinical development. So, you know that the preclinical, it is before the clinical trials. So, preclinical trials which will be done on the animals, and clinical trials which will be developed, which we will be going to do on the human beings. So, the drug development phase has been divided into majorly two steps. One is preclinical and second one is clinical trials. So, here in preclinical and clinical trials, you are going to check your drug on first time on the animals, and after of that depending upon the approval of an regulatory authorities, and then the drug will pass to the clinical trial. Okay. So, this is then a flow chart where you can see here the drug discovery and development. So, from the first slide I am discussing you, the first step was a drug discovery and the second step was a drug development, right. So, in drug discovery, what you have to do here, you have to choose the disease. So, if you do not having any target, or if you do not having a particular disease, so for what you are going to invent the drug, so how can you invent the new compound for a particular disease, right. So, first you have to choose the disease. So, for what particular disease you are going to invent the new drug, and next you have to choose the target, and finally you have to lead the compound. In drug discovery process, what is going to happen here? You have to lead the compound, you have to identify the compound. The lead compound will be get found, and then it should be optimized. The lead compound should be optimized. By here you can be completed the clinical by here you had completed the drug discovery process. Once the drug has been optimized, then the drug development phase will be get started. So, in drug discovery phase, what we are going to discuss here, first you have to choose the disease, you have to choose the target, you have to identify the lead, and you have to optimize the lead, by where you had complete the drug discovery process, and after of that the drug development process will be get started. In drug development process, what is going to happen? So, I had said that we having major two steps in the drug development, one is preclinical and second one is clinical. So, let us see here, first one is we have to go with the preclinical trials, which will be carried on the animals, and after of that you will go with the clinical trials, which will be first time our drug is going to check the activity on the human beings. So, after preclinical trials we are going with the clinical trials, and before going the drug to the clinical trials, you need the approval from the regulatory authorities, which will be discussed, which I will be discussed in the coming slides. And first the preclinical trials, and second one is the clinical trials, and after of that you need the regulatory approvals, and finally the drug will be get marketed. This is the simplified or simply form that which you can understand very easily what about the drug discovery and what about the drug development. So, I will discuss one more time here. The drug discovery is a process where you can invent the new compound by choosing the target or by choosing the disease for which particular disease you are going to invent the new drug. And in drug discovery process, you are identifying the lead compound and you are optimizing the lead compound. Once you had identified and optimized the lead compound, then the drug development phase will be get started. In drug development phase, we are having the major two steps, one is preclinical trials and the clinical trials. So, we will first carry with the preclinical trials and we will send the application to the regulatory authorities, and once the regulatory authority give the approval for our studies, then we will carry in the clinical trials, and once the clinical trials studies has been completed, we are having the four clinical phases, clinical studies, we are having the four clinical trials here, phase 1, phase 2, phase 3 and phase 4, which we will discuss in the coming up slides. So, in these clinical trials, after completion of the clinical trials, again we need the approval from the regulatory authorities, and finally our drug will be get marketed. Okay. So, drug discovery and phases, what are the steps? What are the steps? So, here we will discuss one by one in a very detailed manner. So, what are the steps in the drug discovery and development? So, the first thing what you have to do, you have to discover the drug. So, first you have to select a compound, right. So, you have to discover the new drug. First you have to select the compound which the compound is going to give that such a activity for a particular disorder or particular disease. So, first what is the first step in the drug discovery and development? First you have to select the compound and you have to go with the vitro testing. After you had select the compound, you have to go with the vitro testing, you have to test in the vitro in laboratories, and then you have to identify the lead and you have to optimize the lead identification, which you can call as an lead generation. Also, you are generating the lead, you are generating the new compound, you are generating or you are identifying the compound. So, once the lead compound has been generated, then the lead compound should be optimized, and then you will go with the preclinical development and next clinical trials, regulatory approval, and finally the drug will be get marketing into. So, finally the drug is going to marketed. So, up to here it is a drug discovery, and after that you will see the drug development, right. And so, we will discuss about the things. So, first you have selected the compound, and after selection of a compound, what you had done here, after selection of a compound, you had done with the vitro testing, right. After vitro testing, you will identify or you will generate the lead. So, first we will discuss about the lead identification. So, the name itself indicates lead identification, you are going to identify something. So, what you are going to identify, the name itself, if you see the name here, the lead identification. So, what is an identifier? You are identifying. So, the lead identification, where it refers to identify the molecule which having the activity which you can develop into the drug. So, first you have to identify that a particular drug having the particular activity, and you have to make that molecule you have to develop into the drug. So, the lead identification you can call as any lead generation, and which will refers to identify a molecule or to identify any substances with the activity which you can develop into the drug. So, if the lead molecule do not having any activity, so how can you develop into the drug, right? So, if you have to develop the drug, first you have to identify the lead molecule whether it having the activity or not. Once you have detected that the particular lead molecule having the activity, then you can develop into the drug. And once the lead identification has been done, then you have to go with the lead optimization. So, what is any lead optimization? Lead optimization is nothing but you will it is a somewhat enhancing process. It is the most promising steps by where you can increases the efficacy of any drug, you can improve the effectiveness of any drug, you can decreases the toxicity of any drug, you can increases the absorption of any drug. So, what are the things you didn't done on the identification process, by the optimization of any lead compound, you can increases or you can develop the most promising compound into the market. So, here after the lead identification and after the lead optimization, what you have to do here, you will go with the drug development phase. So, we will discuss about the lead optimization. What is a lead optimization? Lead optimization, what it aims here, it will enhance the most promising compound. So, what it will enhance, what is the most important promising compound which can be enhanced by the lead optimization? You can increases the efficacy of any drug, you can diminish the toxicity of any drug. So, by optimization of the drug, you can decreases the toxicity, whether the drug having the toxicity or what are the condition or what are the parameters you can do by lead optimization. You can diminish the toxicity or you can increases the absorption or any pharmacokinetic parameters in the lead optimization. Once the lead has been optimized, the medical science, the medical chemist, so what we have after the molecule has been identified and it has been optimized, then what we will do here, we will go with the synthetic methods. So, we will go with the organic synthesis method and biotechnological methods, and who will do this organic synthesis method or biotechnological method, which will be done by the medical chemist. So, medical chemist will optimize the drug molecule by using the advanced organic synthesis method or biochemical methods. Okay, next we will go with the preclinical trials. So, preclinical trials which will falls under the drug development phase, which is one of the most important step in the drug development. So, if you see here the preclinical trials or preclinical evaluation, what is the main aim of the preclinical evaluation? You have to make a file. To whom you have to make a file? You have to make a file to the regulatory authorities. What the file you have to make here, what the approval you need from the regulatory authorities, you need the approval. You need the approval for the preclinical studies, you have to file the IND. So, what is an IND? IND which represents the investigational new drug. So, you had invest the new drug and you have to file that invest new drug to the regulatory authorities. So, what is the main aim of the preclinical evaluation? The main aim of the preclinical evaluation, you have to file the IND. To whom you have to file the IND? We have to file the IND to the regulatory authorities after completion of any clinical after completion of the preclinical phase. So, once the IND approval has been done by the regulatory authorities, and now our drug is going to check the activity first time on the human beings, because in preclinical trials we are going to check the activity on the animals, not in the human beings, right. So, we are going to check the activity. So, in preclinical trials we will check the safety of any drug, we will check the toxicity of any drug, we will see the most of the pharmacological actions like pharmacokinetic parameters, pharmacodynamic parameters, the absorption of any drug, metabolism, distribution, how it is excreted from the body, is there any toxicity effect, is there any teratogenic effect, all these effects which you can be done by the preclinical phase, and you will file the IND and you will take the approval from the regulatory authorities. Okay, you had done all the procedures in the preclinical trials by using the animals, you had filed the IND and you have to wait for the approval from the regulatory authorities. Once the regulatory authority has given approval for your IND, then the next step will be started, which is a clinical phase. In clinical phase, here once the approval has been get from the regulatory authorities, now our drug is going to first time going to check the activity on the clinic, which means human beings. After getting the permission from the regulatory authorities, the clinical phase will be get started. So, what are the steps involved in the preclinical studies? So, preclinical studies is not a simple task. Here having a lot of steps, where lot of steps has been involved in the preclinical studies. So, what are the steps involved in the preclinical studies? First, you have to identify the drug target. So, first you have to know the drug target. So, first you have to identify the drug target. What the what is the after you had identified the new compound. So, you have to check the target also. So, you have to identify the drug target, and the next step was to develop a bioassay. So, what is a bioassay? It is a biological assay where you can determine the potency of a drug. So, which will be discussed in your next coming semester in detail about the bioassay. So, here let me see the definition somewhat which will be useful here. So, bioassay is a biological assay where you can determine the potency of a drug in vitro or in vivo methods. Okay. So, you having lot of procedures, you can having lot of bioassays, multiple point bioassay, bracketing bioassay, matching bioassay, interpolation bioassay. So, these are the procedures by where you can check the activity of an or you can check the potency of an drug. Okay. So, we have to develop a bioassay. After develop of a bioassay, you have to screen the drug by using the assay. You have to screen the drug, and after of that you have to establish the ED50 and LD50 of a drug. So, what is mean by ED50 and what is mean by LD50? So, ED50 which represent the effective dose, and what it represent the 50% of a population. So, 50% of population you will check the activity or effective dose of an activity in the 50% of population, and what about the LD50 which represent the lethal dose, which you can call as a toxic dose also in the 50% of population. So, if you are going to discover, if you are identifying or if you are establishing the LD50 or ED50, it means you are checking the therapeutic index of a drug. By the therapeutic index you can understand whether the drug is in a narrow therapeutic index or whether the drug is in a wide therapeutic index. So, which I had discussed in my previous lecture. If you are having any doubt on the therapeutic index, you can visit the. And what the therapeutic index represents here? The therapeutic index represents the 50% population of an effective dose to that lethal dose to that of an effective dose. Here some drugs which you had been marketing, some drugs which you had been invented and which you had been released into the market. Some drugs having the narrow therapeutic index, some drugs having the wide therapeutic index. So, what it represents? If the drug having the narrow therapeutic index, if the drug having the wide therapeutic index, what it represents? So, here I can say you here, if any drug having the narrow therapeutic index, it means it having it means you have to take very conscious or by using of a particular drug. If you slightly increase the dose, it does, if you even you have to slightly increase the dose, it can causes the toxicity action. The best example falls under the narrow therapeutic index was a warfarin. So, if you slightly increase the dose, which can causes the toxicity effect. And what about the narrow? If we have, now we had discussed about the narrow therapeutic index. What is about the broad therapeutic index, in wider therapeutic index? So, after taking of a dose in a very much way, or if you have increased in a little bit manner also, it cannot show any toxicity actions as like the narrow therapeutic index drugs, right. So, first what you have to do here, what are the steps involved? So, let leave about this therapeutic index, which is not very important in this topic. If you are having any doubts here, you can visit my the previous lectures. Okay. So, what about the steps of any preclinical studies? First, you have to identify the drug target, and you had identified, and you have to develop the assay, and after developing of the assay, you have to screen the drug. Once the drug has been screened, you have to establish the LD50 and ED50, which I had said you about the lethal dose 50 and effective dose 50 in sense 50% of a population, and finally you have to file for an approval. These are all the steps which you can see in the preclinical evaluation. Okay. And next, what about the preclinical studies? So, now we had seen about the preclinical evaluation, let me see about the preclinical trials. The preclinical trials you can call as a non-clinical studies, because which you cannot discussing on the human being. So, which you are not going with a human being. So, what you have to do here in drug development process, once the drug development process, the preclinical trial will be get started. Here the drug is going to check the activity on the animals. What is the main aim on the preclinical trials? You have to check the safety of a drug, how much safety the drug to give in the next phase for any human beings. And after what is the main agenda in the preclinical trials? You have to file the IND for an approval. Okay. So, these are all the steps students where you can check in the preclinical activity. You will check the pharmacokinetic parameters, pharmacodynamic, bioequivalence, bioavailability, toxicity studies, reproductive studies, teratogenic studies, and you can see the mechanism of action, mutagenicity, carcinogenicity, immunotoxicity. All these studies you can be done in the preclinical trials. And in preclinical trials we will go with the 4 testings. One is pharmacological testing, and second one is preliminary toxicity testing, and third one is pharmacokinetic testing, and fourth one is pharmaceutical testing. So, what about the pharmacological testing? In pharmacological testing you will see, is the drug showing any hazardous effect, is the drug is showing any bronchial constriction, whether the blood pressure is decreasing or falling, or whether the blood pressure is rising, you will see here. And what about the preliminary toxicity testing? Here you will give the dose for 28 hours and you will check or you determine the non-toxic dose of any drug. And what about the pharmacokinetic testing? Pharmacokinetic testing you will go with the ADME parameters. And what about the pharmaceutical development? In pharmaceutical development you will develop the formulation which is suitable for any coming phase, right? And in preclinical trials you will go with the potential benefits, you will see the safety concern, you will see the best dose of any drug, and you can choose the route of administration, which route of administration you can give the drug. And finally, you have to go with the IND application. IND application will having a certain information what the IND will do here, investigational new drug will do here. They will decide whether the study has been done properly or not. They will see the drug is made or not, who makes the drug, and what the exactly in the drug, and how the stable it is. All the parameters will be checked by the IND. All the parameters will be filed in the IND, and finally we will complete the preclinical phase. Thank you so much.